Thank you for joining us tonight to hear Dr. Kristy Forsgren's talk. I'm Lisa Collins, and I'm the president of SCAS. We're really excited to have you all join us for tonight's event as we spotlight our board members with these talks. We hope these events increase your connection to SCAS and show you the value in continuing to support SCAS and our mission to support the science and scientists working in Southern California. For our guests, if you enjoy this talk, we encourage you to join SCAS. Your membership supports the science and scientists and the work that they're doing in Southern California, and also supports students. We hope to see you at future SCAS spotlights and at this spring's annual meeting, which will be held in a similar virtual format. Information on the annual meeting, including registration and abstract submission, will be live next week. So check back on the SCAS website for that. We'd also like to take a moment to recognize and thank the editors of the Smilodon, which is our newsletter. Editors Brad Blood and Scott Graff are stepping down after many, many years of service to SCAS. Brad Blood is a former SCAS board member and a past president of SCAS, and Scott Graff is an accomplished vocalist and voice teacher. Both Scott and Brad work for Somas, which is a full-service consulting firm, and for many years, Somas printed the Smilodon for us. SCAS is grateful for the generous contributions from Scott, Brad, and Somas. So it's my pleasure tonight to introduce Miss Bonnie Lay, an alumni of the SCAS Research Training Program from 2009 and 2010. She presented at the SCAS Junior Academy of Sciences meeting in 2009-2010 on her research, discovering and describing a new species of sea slug in the Caribbean. She studied organismic and evolutionary biology at Harvard and was an inaugural Schwarzman Scholar at Tsinghua University. Previously, she researched climate adaptation of endangered penguins in South Africa and helped initiate Wildlife Conservation Society's Marine Conservation Program, Myanmar. Ms. Lay is currently the head of Global Strategic Partnerships for Microsoft's AI for Earth program. We're thrilled to have her join us tonight as the moderator for this talk. Thank you, Ms. Lay.
Thank you for the kind introduction, Lisa. I'm so excited to get to come back and join all of you virtually. The research training program was so fundamental to helping me get started as a scientist, and really is what helps me lead me to my path today, getting to work on applying machine learning approaches to work on environmental conservation. And it is my great pleasure to introduce the speaker for tonight, Dr. Kristy Forsgren. Kristy Forsgren is an associate professor at California State University Fullerton. She received her Bachelor of Science in Marine Biology and Master of Science in Biology at the California State University of Long Beach, and her doctorate from the School of Aquatic and Fishery Sciences at the University of Washington, focused on fish physiology and endocrinology. Currently, her research is focused on understanding the reproductive biology of a variety of fishes. She has investigated many unusual fishes, including weedy seadragons, oarfish, mola sunfish, and salmon. She has been a SCAS board member since 2016, and today she will be sharing with us her research on surfperches, a group of marine fishes that give live birth to offspring. Thank you so much for joining us, Dr. Forsgren. I'm going to let you take it away. And for all the attendees on the line, if you have any questions throughout, feel free to use the Q&A box at the bottom of your screen to type in your questions, and we'll get those answered at the end. Thanks again, and Kristy, take it away from here.
Great, thanks, Bonnie. I want to thank everybody joining us tonight to tune in during our Valentine's Day week to talk about some of the most intimate reproductive structures of fishes. So I just kind of want to start off with giving you a little bit of background about fish and why I find that they're so interesting. They have incredible diversity. 71% of our Earth's planet is covered by water, and regardless of what that water composition is, you will always find fish there. So our water provides habitat to over 30,000 species of fish, and they account for about 50% of all vertebrate species on our planet. Fishes have this amazing diversity and adaptive radiation in this aquatic environment and be able to live in oceans and lakes and rivers. They live at the abyssal depths of the ocean. They live in ponds up into Tibet. They live in very, very voided areas with no salt, so almost distilled water in the Amazon, to very, very saline waters. So they're really amazing, can be found just about anywhere. And in addition to that, they have a wide variety of body forms, which is also quite interesting. But for me, that fish diversity really comes into play when we talk about reproduction. If you can imagine any kind of crazy reproductive strategy, you're going to find that in the fishes. And reproduction is really important to understand in fish, especially with climate change occurring, with how we utilize fish as a food resource, and how we just enjoy them for recreational purposes as well. Nothing is as diverse as reproduction in the fish. We have everything from a parasitic male in anglerfish, mass spawning like in the snappers and the tuna, direct copulation, and even animals that change sex like our local California sheephead. So you might be wondering, well, how do fish reproduce? The males have testes, just like males of other vertebrate species. This is a picture of a blue rockfish testis that we've dissected, and just showing the different seminiferous tubules in which sperm development is occurring. So the fish will produce sperm, and they will either be released into the environment or into the female's reproductive tract to fertilize the ova. So again, just like other vertebrates, fish have ovaries. This is a blue rockfish ovary and a dissection of a zebrafish that has been cut through paraffin histology, where you can see the different stages of ova. So the development of the sperm and the testis is very similar to what we see in other vertebrates, which is quite interesting. Everything else about them is quite unique. The egg of the fish is really interesting in that it has what's called a micropyle. And a micropyle is a hole. The hole is where the sperm must travel through in order to fertilize the egg. And a lot of people don't know about this micropyle. We're used to just regular fertilization where the egg attaches and penetrates through the egg. For the fish, they have to find this special little trap door to go into. So different types of reproduction in fish that we see are broadcast spawners. These are the fish that just gather together in groups. They release the egg and sperm into the environment. There's millions and millions of unprotected gametes. And you just hope by fate that some of these eggs get fertilized, that these larvae are going to develop into sexually mature adults one day to propagate the next generation. Of course, the strategy behind producing millions of gametes is they're relatively inexpensive for the animals to produce, and it kind of satiates any predators. So we see this a lot in a lot of marine organisms, like here I've shown the snapper, and we also see it in the salmon. And here's some nice little salmon embryos that are starting to hatch out. Sperm can also be directly delivered into the female's reproductive tract. And this is really the area of study that I'm most interested in. About half of the fish that exhibit this internal fertilization are freshwater, leaving the other half, about 2% of all fishes, that have internal fertilization in as a reproductive strategy. And even though there's half of them are considered to be freshwater and half are marine, we know a lot more about the freshwater internally fertilizing fishes. With our oviviparous fish, sperm is going to be delivered directly into the female's reproductive tract. The eggs are fertilized. They have a large yolk content. Then the female just serves as protection for them. After a while, she's going to actually give birth to millions of larvae, which will then develop in their environment. So an example is one of our local rockfishes. Here's a blue rockfish. Here is her ovary. I know it's a little bloody and gory. You get used to this, believe me. But if you look really closely, and hopefully you can see this little golden shimmer, these are the eyes of the larvae that are about ready to, or would have been born. There's also viviparity in the marine fishes. And in this situation, the sperm is introduced into the female reproductive tract, and the young develop some type of connection with the maternal tissues. And it's the female's tissues that help to nourish the offspring, very similar to a placenta in a mammal. We don't call this a placenta. It doesn't have quite the same type of connections, but at any rate, we see that here's a female black perch where we've removed the body wall. We have dissected out this little sac of babies, and you can see how vascularized that tissue is. That tissue and that vascularization are providing gas exchange for the developing young and any nutrients. And in fact, we found that each little fish has its own little sac. And something I won't really have a lot of time to get into and talk about today with the females, because we're focusing more on the males, but I have a student who is currently doing CT scanning, and we're trying to elucidate the female reproductive tract and the tissues connecting the embryo and the female. So these little green lines represent all of the different individuals developing within the female's ovary, which is bordered by this purple line that you see there. So I'm going to talk to you today about the reproductive morphology of surf perches. And I would just be ridiculous in saying that I have done any of this work all on my own. There have been so many students have greatly contributed to this research. They come up with wonderful ideas, they conduct interesting experiments, and it's the work of all of these students that I'm compiling what they have discovered and what we have found about these fishes that I'm presenting to you tonight. So the surf perches belong to the family Ambiotocidae. And there are two subfamilies, the Amphistykinae and the Embetocinae. So the Amphistykinae has much fewer fish, kind of characterized by this walleye surf perch, while the Embetocinae are a much larger subfamily, and they include the surf perches like the black perch. So I want to tell you a little bit about the reproductive structures and what is known in the literature before we started studying them. So in the Amphistykinae, they actually have what's called a bony plate. So this is located on the anal fin. So here my drawing of a surf perch, you'll see this little blue square. And right here, this is the anal fin. And located on both sides of the anal fin is this bony plate surrounded by a fleshy tissue. And this right here is a ventral view of that. So the head would be on this side here. This is the anterior side, posterior side. Looking down here, we have the bony plate and the fleshy structure associated with them. Now, back in 1952, it was hypothesized that these bony plates were used to grasp onto the female and hold her into place during copulation. Fair enough, right? Also, in the Amphistykinae, a genital papilla had been described. And so again, on this side, we have the anterior region of the fish, posterior region over here, and we're looking at the ventral side. So imagine that you pick this fish up and you've turned it so you're looking at its belly, and it has a genital papilla. A genital papilla is the tissue in which the genital material would exit out of. So this was believed to be the mode of sperm transfer in the fishes. And even though it was only looked at in one species, it was believed to be a family characteristic. If we take a look at the Ambiotocids, they've also had one species, the shiner perch, in which this genital papilla has been described. Again, it's been hypothesized that this is the mode of sperm transfer. However, the Ambiotocids also have what's called a flask organ. So again, on this anal fin, they have a paired structure. It's a fleshy structure located on both sides of the anal fin. And this was sometimes referred to in the literature as being the mode of sperm transfer and introducing the sperm into the female's reproductive tract. But most of the literature says that the function of this flask organ is completely unknown in the Ambiotocinae. So the surf perches display a very, very interesting type of reproductive behavior. So this is a video shot by one of my grad students, and we just happened to be at the Cal Academy of Sciences, and these surf perches started this mating behavior. Now, this mating behavior and copulation in the wild has not really been well observed, and it's not been well documented. But as what we see here This one that's dancing around is the male. Here's the female looking on at the male, wondering what's going on and what is he doing. She's clearly watching him. And if we give them just another couple of seconds, we have what we believe we saw was an actual copulation. So they go through this elaborate dance. I promise they're getting there. Almost there. It's super quick. Watch, watch, watch. Boom, done. That was it. That's the copulation of the surf perches there. So the copulation is not well documented. From that little second that we saw, it's very difficult to determine how the sperm was actually transferred to the female in that very brief contact. So this mode of sperm transfer, quite unclear. And is it the papilla or was it the flask organ that was involved in transferring that sperm? So we've started out by collecting fish. We use beach seines and we collect surf perches on local sandy beaches in the area. And during COVID, we've gotten very creative and we've started utilizing museum specimens. So we're looking at those while we've had limitations of working in the lab and working in the field. And my students are so dedicated to these projects. They've taken these jars of stinky fishes home and they've taken my microscopes home and they're still working despite all of this. So we usually dissect the tissues. Here we have, we're examining them under microscopes and dissecting scopes, trying to learn about them. We take photography. We do paraffin histology where we embed the tissue into paraffin wax, section it into slides and examine those. And we've done a lot of clearing and staining so that we can see the fleshy and the bony elements of the anal fin in particular. So let's start off with the genital papilla. So this is one of the regions that had been described as being present in the shiner perch. And so here we have the genital papilla. Again, we're looking ventrally on the animal. So over here would be the anterior end towards the head. Back here, this is a posterior end towards the tail. And we've just flipped the animal underside to take a look at what's underneath there. We have the genital papilla. And we have a flask organ here. So we have the anal fin, we're looking down. You can see that fleshy tissue really well here. I promise you there is fleshy tissue on this side as well. We're taking the same fish and we're looking at the left side of it. Here again would be the head and over here would be the tail. And so we're seeing the genital papilla here and the flask organ is over here. And these fish also have this little curlicue, which we'll talk about a little bit later. So even though it was said in the literature that only one of the Amphistechini had been examined and had been identified as having a genital papilla, we have actually looked at 4 of the 6 Amphistechini species, and every single one of them has this genital papilla. So this is their little genital structure. And we have found that in 4 of the 6. We just haven't been able to find representative species of the other 2 of this subfamily. When we took a look at the Embiotocini, we have looked at 17 of the 19 Embiotocini species. Every single one of them has a genital papilla. So we are working to describe them. They're all very diverse. They look very different. We do feel like that they could be species specific. So we really wanted to get at this question as well, what is the genital papilla and is it really involved in transferring sperm to the female? So what we did is we got some fish into the lab and we dissected from the genital papilla, which is that external opening on the ventral side of the fish. And we just started following it through and dissecting out the structures. So what we found, here's the paired testis, and this is the sperm duct, which leads directly into the genital papilla. Here we have the urinary bladder, and the urinary bladder is closely associated with the testis and the sperm duct. So we see that the urinary bladder and the testis join together in the genital papilla. So we have this direct connection. We have seen this in all of the surf perches that we have dissected. So quite the diversity in the testis structure. Here's the testes going down to the sperm duct and the genital papilla. We've removed the urinary bladder on a couple of these. Again, here's the testis, the sperm duct, and this external genital papilla. Same on the walleye. The testis, here's the urinary bladder. They're both joining together into that genital papilla, which is going to exit the contents, either the sperm or the urine, into the external environment. We really wanted to verify this, so we did some paraffin histology. So we dissected the tissues, embedded them in paraffin wax, we sectioned and stained them, and now we're able to take a look. And sure enough, as we expect, in the testis, and this dwarf perch is the best one because it's the most mature, we can see these mature sperm within the testis. Part of the sperm duct is missing here. It's a very small structure and just got missed here in this particular section, but it would connect. And right here in the sperm duct are all these mature sperm, and it is going directly into the genital papilla. So we have been able to verify in several ways the testis is directly connected to the papilla, and this is likely the way that the sperm are exiting from the male's body. So just one more look here at it. There's our testis, the sperm duct, and our genital papilla. We see in the sperm duct of this particular animal, this walleye that we did paraffin histology on, the sperm duct was packed with mature sperm. And so this is just a picture of the histology of the multiple stages of sperm development in the surf perch. So we can see some primary and secondary spermatocyte development, spermatids, which are little tiny, they've undergone their meiotic divisions. There's 4 of them from those divisions. They start to develop their tails, which you can see their tails stain really pink. They start to congregate together. So in this outside circle, the heads of the sperm gather together with the tails inside. And as what they start to do is form what we call a spermatozygomata. And the spermatozygomata is just a large collection of the sperm that will all be transferred together. This is going to ensure better success of fertilization. We wanted to take another look at that papilla because it seemed very interesting that the urinary bladder and the testis were so closely connected, and they both appear to be going into the genital papilla. So this is the way that you want to orient yourself with this animal is imagine that the fish is tail end facing you, head end going away from you, and this is the ventral side. So as what we did is we took that genital papilla off and we did cross sections of that to look at the lumens within there. So is what we found. So here is the body wall of the fish. You can even see the spine rays here of the fins. This small structure is actually the urinary duct. Also, we find here the sperm duct is having this exit through this lumen in the papilla. So the papilla is this entire structure. So while it has been called a genital papilla, that's actually incorrect. These fish have a urogenital papilla. This is an exit point for both the urinary product and for the sperm. So let's talk about the flask organ. It has been thought of as being the intromittent organ, and the intromittent organ is the structure that is inserted into the female reproductive tract and transfers the sperm. So now that I just showed you that surf perches have a urogenital papilla that's directly connected to the testis, how in the world is this flask organ involved in reproduction? So here we have a black perch, and again, the flask organ is located on the anal fin. You can see a little bit larger structure of it here. This is a paired structure. So looking down again on our fish, we're looking at the ventral side. Here we have the anterior end, the head would be here, and the tail is here, this posterior end. So looking down at it, we see the flask organs on either side of that. So they are paired structures. This is a clearing and staining so that you can see the pink stains the bony elements. The blue color is more of the fleshy tissue. So we're looking at a side view here. This is just orienting you to the fin. This is an anal fin spine. Again, over here, we would have the anterior or head side, whereas over here, we're looking more towards the posterior side of the body. And we see that there is this tubular structure, and outside of that is a tubular sheath. and that tubular structure is housed within that sheath. So looking at the same fish ventrally, here's our anal fin spine. This is the tube. And I will tell you, this tube does have a lumen through it. And then we also can see that we have this tubular sheath that is surrounding that tubular structure of the flask organ. So one of the things that has been noted by many people is that when you apply pressure to the flask organ, it does indeed come out of this little pocket, this little fleshy sheath that it is in. Quite fun to play with, I must say. And I will mention that when you do this, there is a mucus material that does come out of it, and that will become important in just a little bit. So we've dissected off the flask organ. Again, this is the tubular structure, and it's housed within this tubular sheath. We wanted to get a close look at the lumen. So we did the histology, and this is the exact same animal tissue. We just cut it right down the middle. Here you can see that there is a lumen within that tubular structure, and there is a lumen inside of the tubular sheath where it goes in and fits. Further back on the flask organ, you can see that there is a little bit of space, but we're really looking at more fatty tissue here, where we don't have that clear lumen here where that housing is occurring. So we have looked at the 17 of the 19 embryotocini that we have. Every single one of them has paired flask organ structures. Again, they are incredibly diverse depending on the species. So I've tried to highlight them here with these little boxes so that you can see them. Again, they have one on either side. The shape and the size does seem to be very species specific. I have 2 students right now who are doing geomorphic morphometrics on the genital papilla and the flask organs so that we can statistically analyze how different or similar these structures actually are on the fishes to determine if they may be species specific. So what about those amphistechini? They don't have the flask organ, but remember that they had a bony plate. When we started looking more closely at the bony plates of the amphistechini is what we found. So there is a bony plate located here, and that bony plate is composed of bone, just like the fin rays or any other bone structure within the fish. Is what they also have, though, is a fleshy structure associated with them. So this is the side view. And then here we have the ventral view looking down. You can see the fleshy structure on top of the bony plate. So the bony plate is here, and then there's this fleshy structure. And we found this in all 4 of the species that we had access to. So 4 of the 6 have this bony plate as well. And it's been thought, and at some point in 1952, Tarp actually said, well, he recognized that there was this bony plate, there was this flask organ, and he said that they were very similar in nature of the tissue. So we've been kind of looking at, are the functions of the bony plate and of that flask organ very similar? One of the things that we've noticed when we've been collecting the surf perch is that we only see the flask organ fully developed during the breeding season. So in this case, we're looking at a black perch. So as what we find is that in the winter and the spring and the summer, we have a lot of individuals that don't have any flask organ. We can see where the tissue had developed there, but it has regressed and is no longer present. So we have staged the development of the flask organs as no development, intermediate development, where we could start to see the flask organ developing, and full development is a score of 2, where we can see the tubular structure and that little tissue sheath surrounding it. So here on our y-axis, this is the percent of animals that we have found that have these different stages of flask organs. So in winter, we actually see that we have all 3 of them present. In spring, we see that there's either no development or some intermediate development. Summer, we have all of them. In fall, when these animals are actively reproducing, all of the animals that we have collected have a fully developed flask organ. On our z-axis here, I've shown you the gonatosomatic index. And the gonatosomatic index is where We dissect out the testis, we weigh it, we weigh the entire body, and then we take this index of how much of the total body tissue is dedicated to the testis. And this is our gonadosomatic index. It tells us how much tissue within the testis is being dedicated to sperm development. And it's what we find in the fall is we have this really high gonadosomatic index, which tells us not only are we seeing the flask organ development seasonally, but it does have something to do with reproduction. We don't see the flask organ fully developed typically in the non-breeding season, but every male has that when they are actively breeding. And I don't have a slide to show you this, but I will tell you we've also noticed the same thing with our bony plates and the amphistechini. The bony plate, because it's a bony element, is always present. But around that, there's this fleshy tissue. And that fleshy tissue will regress in the non-breeding season. And then we see that fleshy tissue has recrudesced, has come back during the breeding season. So we've also taken a look at the histology of the flask organ development based on these stages. So again, this is going to be a ventral view. You've taken your fish, you're looking down on it, and we're going to do sections longitudinally to take a look at that. With no development, we can't see any development of the flask organ. So here you can actually see the bony elements of the body. We have an intermediate development where we're starting to see this flask organ develop. Then when we get to the full development, you can actually see these lumens inside of the tube and around within that sheath where the tube is housed. Again, it's corresponding with gonadosomatic index. When we're actually seeing that flask organ develop is when we have the highest tissue mass of the testis. So we really wanted to get a closer examination of what are the relationships between the testis, the genital papilla, and the flask organ in these species? We've looked at them independently and in a variety of ways. So we have taken a look at them externally to see that sheath, and we see the flask organ, the tube here. We've looked at the histology. What we've started to do is CT scanning them. So we scan the anal fins, and you can see that this is where the tube is coming out, and this is the soft tubular structure around it. With these CT scans, we've colored and segmented, basically taking all of these structures from the CT scan and coloring it so that we can make a three-dimensional model. So over here, we have the testis in blue, the urinary bladder is red, the genital papilla lies below that in orange. This blue structure is the whole anal fin. The green here is that tubular structure of the flask organ. And this aqua color is that tubular sheath. So it's what we do is we make this three-dimensional model, and then we can actually rotate this model and take a look at how these tissues are oriented with one another. So let me stop it for you right here. If we're looking at the animal, and now we're looking if the head was facing towards us, but looking down towards its genital area. We see the testis here, and the red again is that urinary bladder. Remember I said that they're very closely associated with one another. It almost appears as though the sperm duct goes through and is very closely associated with the urinary bladder. And in other parts of the scan, if we go into the scan and don't have the whole three-dimensional model, we can see that the sperm duct and the urinary duct do enter into the genital papilla. The other thing I want to show you on this rotation, let this go through here. If we stop it right here, and now we're looking, if the head is away from us and we're looking at the tail region and we're looking into that flask organ on both sides of this anal fin, so here's the anal fin, we can actually see that tubular sheath structure. And inside it, that green, that is actually the tube that's being housed inside of it. So now we know that there's this close relationship with these paired structures. We know that it's seasonal, and we still wanted to know more about this tubular structure. So what we did is we took some of these tubes and we did scanning electron microscopy on them. And so here is a dwarf perch, there's the tube, there's the structure around it. The same structure here. This is the tube, and then this is the tubular sheath surrounding it. When we did scanning electron microscopy, is what we found on the tube were all of these little micro ridges. Now, micro ridges are really interesting. They are present on epithelial tissue in which mucus is held. So it's thought that the structure of these micro ridges helps to maintain the connection between mucus and that epithelial tissue. So remember, I told you that when we apply pressure to this tube, a mucus tissue actually comes out of it. So we think that this mucus may come out of here, and the micro ridges located on the tube are helping to retain that mucus on that particular structure. So why would an animal do that, right? We think that there might be some type of a pheromone or some complex carbohydrates or proteins that are being released in this mucus that might cue some of this reproductive behavior that we have seen in these animals. So what is it doing? We think that we might have some tissue, this soft tissue here that's surrounding. Remember, I told you we'd come back to this little squiggly structure on our shiner perch. We think that this is the structure that might be producing some of this mucus. And we have this mucus associated with these tubular structures in some way. We still don't know what that is. We're getting to that point, but we do want to collect this mucus and take a look at it and see if we can do a chemical identification of it and determine whether it might be some type of a hormone, a protein, a carbohydrate, something that could be used as a pheromone for communication. It's very clear that the fleshy tissue of the bony place of the amphistechini and the flask organ of our embryotoceny are related to reproduction. We only see them when they're breeding. So there might be something associated with that pheromone communication. So what's the big takeaway message other than you got to see a lot of fish genitals tonight? All of the embryotoses, and I say all, of the 19 of the 23 species that we have been able to take a look at, all of them have a urogenital papilla. That urogenital papilla is most logically and likely the mode of sperm transfer, the way that the male is transferring the sperm into the female's reproductive tract. We never saw sperm in the flask organ. We've never seen sperm located and associated within a bony plate. So we don't think the flask organ is involved in sperm transfer. Doesn't mean that we don't think it has a very important role in reproduction though. And we found in the 15 of the 17 embryotoceny we've looked at, they have flask organs. 4 of the 6 amphistechini have bony plates. All of these species that we have collected in the wild, they're only present during that breeding season. So they have to be involved in seasonal reproduction in some way. So overall, yeah, surf perch reproduction is really cool and really interesting. I'm going to show these guys mating to you one more time, and you can watch this. So during this part of mating, we hypothesized that this could be a potential time point for the male to be releasing some type of mucus that can communicate with the female, and then they end up having this mating courtship that results in a copulation at the end. But we still have a whole lot left to learn. We still don't know how direct copulation is occurring and how the sperm is transferred directly into the female's reproductive tract. We still don't know what that chemical composition of the flask organ is. There are other fishes that have a similar fleshy structure on their anal fins or their caudal fins that have been shown to be involved in reproduction and produce these pheromones, which is why we think that that's a hypothesis that we should lead on. So we need to take a look at that chemical composition of the mucus that we have found. And we really want to understand this recrudescence of the flask organ. So it appears in the breeding season and it disappears when they're not breeding. So our idea is, because we have this tight association with the presence of the flask organ when the gonatosomatic index is greatest and when mature sperm are being produced, that it's very likely that the growth and the recrudescence of the flask organ tissue in the embryotoceny and the fleshy tissue surrounding the bony plate and the amphistechini is going to be dependent upon androgens. So we believe that the androgens may be promoting that recrudescence and growth of these structures that may be producing a pheromone to aid in communication. And so we're working on that. I have several students in the lab that are working on these different aspects of what we do not know and what we're still trying to understand about this unique group of fishes. So there are so many people to thank. There have been so many students who have contributed to this project. We've also had a lot of assistance with Fish and Wildlife and the Johnson Lab sharing fishes with us because we want to get as many fish and as many different species as possible to examine. We have looked at museum specimens from the Los Angeles Natural History Museum, and the Scripps Ichthyology Collection. This research has been supported by Cal State Fullerton, also CSU Coast, and even some NSF support for this. So at this time, I'd like to thank you all for staying tuned to learn about fish genitalia, and I would be happy to answer any questions that you may have.
Thank you so much, Dr. Forsgren. I feel like this is the perfect talk post Valentine's Day for us to understand the love life of some surf perches. So for all of the attendees, make sure that you drop your question into the Q&A box. So you could open that up and type your question in there, and we'll be sure to read those out loud and pose them. All right, so Dr. Forsgren, I'm going to go started right away and start posing some of these questions to you. From Maria, we have a question that says, aside from seasonal differences in flask development, is there a size or age component to the development? And if so, when do male surf perch begin to develop?
That's a really good question. We have not seen a lot of juvenile fish. When we've done the majority of our collections, we are finding sexually mature individuals. All of the sexually mature individuals have a flask organ. In the few juveniles, and those would be the ones that are not sexually mature that we have found, there is no flask organ development within them. So this is possibly a secondary sexual characteristic that develops at the time of puberty when the animals do become sexually mature. And that also confirms our hypothesis that androgens may be involved in this tissue structure and upregulation of the tissue for combining and recrudescing during the reproductive season.
Fantastic. And a question from Lisa, who says, you showed a great video of the perch at the aquarium. Do many other fish species have similarly fast copulations? That video made it seem super speed.
It's really fast. I mean, a lot of animals will spend a lot more time with courtship and mating than they do the actual copulatory event. And I say that most fish, although last night I have to disclose, I watched a pair of rhinos mating and they mate for 45 minutes with that copulation. So yeah, this is really fast compared to a lot of animals. We don't know is the short, is like really the true answer there, I guess. Observations of copulation events with surf perches is pretty rare. It's very anecdotal. We just happen to be walking by this aquarium. So it's clear that it can happen in aquaria. Whether people notice or know what's actually happening is probably avoiding that documentation. But at least for this particular fish that we saw, it was very, very quick. But they definitely spend a lot more time with the courtship and mating. It's likely to be like that with every one of them, but we have to be able to watch more of them to be able to classify that. Very good question. Don't know, wish we did, but I'll tell you, when we were at the Cal Academy of Sciences and saw that mating, we were on cloud nine for days. We were just so thrilled that we had the chance of seeing that.
That's incredible, and so lucky with the timing. Do you have any collaborations going on with other folks who are helping you try to document more videos of the mating behavior happening with surf perches?
No, we don't. That would be a great citizen science project to have people go out and document any copulations that they see. But right now we haven't expanded that far.
Great. All right, here's another question from Gordon, who says, besides surf perch, are there any other fish that have structures that are analogous to the flask organ and the urogenital papilla?
Yes, there are a lot. So we see a lot of these reproductive structures in freshwater fishes, but yes, we also see them in our marine viviparous fishes. So I have had students who have studied urogenital papilla of rockfish. They have the same role where there's urinary and reproductive materials that exit the urogenital papilla. So we do see urogenital papilla are very common on those animals that do have copulation as a form of reproduction. However, I will say also, I've done some work on our local California grunion. They are not a viviparous species. They do not have internal fertilization, but the male California grunion also has a urogenital papilla. And our research on that particular fish, we found that if you're not familiar with California grunion, the female burrows a hole in the sand. She'll lay her eggs, and the males surround her, and they release sperm into the hole to fertilize the eggs. That sperm is released by a genital papilla, and it's muscular and it has the ability to kind of move. So we think that it is a mechanism for the male to have pressure built behind the sperm to get it down to the bottom of the burrow that the female has dug, and also directionalize where the sperm is going. So there are actually many species that have these urogenital papilla, and the whole idea is whatever, if it's like the grunion, which is technically a spawner, or our viviparous fishes with internal fertilization, the whole goal is to get the sperm closer to the eggs for fertilization.
That makes sense. We have another comment, a question that says, I wanted to ask, does the same specificness also correlate for the bone plate?
So the specificness is to, I'm guessing this means to the role of the flask organ. We think so. So that bony plate doesn't regress and recrudesce because it's a bony element, but there's a fleshy tissue around that. And since we also see that that fleshy tissue is only present during the breeding season and it's absent in adult mature males during the non-breeding season, we do believe that there could be a very similar role there. The bony plates are rather small. I'm not sure that they're used for grasping the females, but that fleshy structure could be similar to the fleshy structure we're seeing with the flask organ, and that it is producing some type of a mucus that has some communicative role with other individuals that aids in reproduction.
Great. And we have a comment from Jasmine that says the future chemical composition project sounds exciting. I'll tag on a question to that. What will that experiment entail? What are your students working on to be able to better understand the chemical composition of that mucus?
Well, I'll say right now, I don't have a student actually working on that project. So if anybody's interested in a master's degree and has some biochemistry experience and interest, that they should contact me. Is what our idea is when we collect fresh, sexually mature males and we apply pressure on the flask organ, we get a mucus material that's secreted. So we want to take that secretion and run it on an LMS, and you can get a chemical profile. So anybody who's taken organic chemistry, remember you run those chemical profiles to see what they are. That's what we want to do with this mucus. By running the chemical profile, there's many pheromones and many hormones that have already been characterized by basically their footprint of that chemical sequence. So we're hoping that if we could run that on an LCMS and get that biochemical analysis, that we might be able to figure out what is the active chemical involved there.
Great. So for all the attendees listening, anybody who wants to participate in that or participate in citizen science recordings of the surfperches, get in touch with Dr. Forsgren. Awesome. Here's a question from Marissa. Does the sperm of the viviparous fish have a shorter viable period than spawning fish?
That's a good question. Not really sure. I don't know that the viability or how long the sperm are viable has been studied in viviparous fishes. The freshwater fishes that are viviparous are more heavily studied, and I don't even recall reading about that. It doesn't mean that it doesn't exist out there. For most egg and sperm, there is a very fine time point when fertilization must occur. So as an example, in humans, once that ova ovulates, there's a 24-hour period of time that it can be fertilized. So typically it is the ova that has a shorter viability or span of time that it needs to be fertilized. Sperm in mammals can actually survive in the female's reproductive tract for several days. So if I had to make a guess, I would say that there is some type of longevity to the sperm that is going to be longer than the egg, but whether there's a difference between internal and external fertilization, I'm not sure.
Or still remains a mystery. And here's a quick, hopefully easy question from an attendee who joined a little bit later, and so wanted to clarify: is the flask organ gender specific?
Wow. Yes, I may not have actually clarified that. Yeah, we were talking about males in the surf perches. So I'll clarify with our marine surf perches, the flask organ is only seen on the males. So we don't see any similar type of structure on the females. We have done some work where we've done the clearing and staining, where we look at the bony elements and the fleshy tissue. Remember those beautiful pink and blue pictures that I had? And we don't really see much differences between the anal fin structure of males and females, with the exception that the males, when sexually mature, do have that flask organ present. So we think that that is species specific. The genital papilla also appears to be species specific and really present on the males. Clearly, the females must release their ova and eventually like their offspring and release urinary products in some way, but it is not in a papilla like we see in the males. That's not true for all fishes. The tide pool sculpins, there's a lot of them that exhibit copulation and internal insemination of sperm. And with those, we actually see that many of the females have a genital papilla very similar to the males. So this is something that is can be gender specific or species specific. I would say with the tide pool sculpins, we see females with some of these accessory structures. With the surf perches, that papilla and flask organ is limited to the male.
Great, thank you for that clarification. And here's a question from Gordon. What is it about the ecology of surf perches that makes viviparous reproduction advantageous?
Yeah, so that's a good question. We want to do some principal component analysis where we look at reproduction and we look at viviparity and habitat and try to get a better understanding of that. So with reproduction, especially reproduction of fishes, and even more so with viviparous fishes, there's not much known about them. I think that with any type of internally fertilizing fish or organism, you see that there is a benefit gained with the female protecting the embryos inside of the body cavity. And then, of course, this would be a little bit different. Seahorses do it similarly, but it's the male that is protecting the offspring. So you have this internal body cavity that is harboring the developing offspring for the form of protection. So predation might be very high in these particular habitats that these animals have evolved this particular mechanism of reproduction. The other benefit to having internal fertilization and having the young develop within the body, not only do you get protection, but you are able to produce fewer offspring, which could seem like a negative, but when your offspring are born, they're larger, they're more mature, and theoretically, they're less prone to that predation. So habitat, we do think habitat might have something to do with this. And in my lab, we are starting to study the tide pool sculpins as kind of a proxy of how viviparity has evolved in marine fishes. In the tide pool sculpins, we can see everything from spawning, where males and females release the gametes. They may spawn in a nest. So eggs and sperm are laid in a nest, and then the gametic association occurs within the nest. We also see some very strange things where we have sperm inserted into the female's reproductive tract, but the female's reproductive tract is able to keep sperm and ova separate. And then she lays both the eggs and the sperm in a nest where we have gametic association external out of the body. And then finally, we have true viviparity, where there is an insemination of the sperm. We have gametic association within the female's body, and those embryos develop to a particular stage before they hatch. So we are looking at why this is occurring. So the tide pool sculpins might give us some insight into the evolutionary benefit of that. And then we'll kind of go back and tease apart that habitat aspect of it.
That's great. Exciting future directions for research. And I'll end us with this question here, which is, what's the final goal with the study? What is the benefit of doing this research? And I'll tag on my sort of corollary to that, which is, I remember you mentioning at the beginning of the presentation that understanding the reproductive biology of various fish species is actually really important to protecting these species and conservation. And can you speak a little bit more about how exactly that would help?
Yeah, so understanding reproduction is the basis of how we get the next generation for a species. If we don't understand reproduction, we don't understand the stochasticity of it, how can we understand our ecosystem? How can we understand how species interact with one another? How do we understand how a species goes about producing that next generation? And we see a lot of aspects of human life that impact animals, and not just marine animals, all animals, where you have endocrine disrupting compounds, you have climate change, all of these things can impact reproduction. If we get a better understanding of it, that really helps us understand what's happening with our populations on our planet. Trying to get back, that's like the big pretty goal, right? What's the actual goal of this study? To learn more about viviparity and marine fishes. This is probably not going to cure cancer. Well, actually, I can say absolutely, this is not going to cure cancer. It's not going to change anybody's lives per se. I get this a lot, and I actually get a lot of criticism at professional meetings for this type of research because it is very descriptive. We're looking at the basic basement platform of reproduction in a fish in which we know very, very little about. You have to start at the bottom. You have to start somewhere in order to understand that. I guess the bigger picture of what I would hope us to understand is just that. Why has viviparity evolved? Why do we see this in a large number of vertebrates? Why do we see it in such few marine species? Why do we see it in few freshwater fishes as well? The really cool thing that I think about this research and why I think that this is not only interesting, and it is important for basic biology. You look at a lot of research that goes on, and it can be any subject, anywhere from cellular biology, molecular aspects, population biology. Many of these systems have been very well studied. And when students come in to study these, they're doing a very, very small part of a project, and they may or may not ever really understand how it fits into the big picture. This type of research, where we're looking at basic biology and basic reproductive function, is really beneficial to students becoming scientists and understanding how the scientific method works, because they get to see firsthand true research and science evolving that has never been described before. When we started this project, we thought for sure, just like everybody else, that flask organ transfers sperm. And every time we make a discovery, we're incredibly surprised by what we find, and it leads to more questions and more understanding about a species. So even though we're not curing cancer here, I believe that the work here is very imperative to understanding fishes and viviparity in vertebrates. And probably a much greater role is that I'm training the next generation of scientists to ask really good questions, form hypotheses, and come up with unique methods and techniques to solve and answer different questions.
Incredibly powerful way to help us close, Dr. Forsgren, emphasizing the importance of basic science, foundational science, and also the training of the next generation of scientists. So again, a huge thank you to you, Dr. Forsgren, for presenting to us today and sharing your research. And also a thank you for all of the attendees who tuned in tonight and hopefully learned something new. I know I definitely did. And I will point you to check out the chat box where there's a posting of the SCAS website. Please be sure to take a look to learn more about the Academy as well as the programs that SCAS supports.
Great. Thank you all for attending.
Thank you so much, everyone. Have a great evening.